Hello, and welcome to the Global COVID Lab meeting. My name is Sabrina Welch, and I'm the Director of Programs and Operations at the Human Vaccines Project, and I'll be your moderator for this webinar series. The Human Vaccines Project is a nonprofit global consortium. Our mission is to decode the human immune system and accelerate the development of vaccines and immunotherapies across major global diseases. The project brings together leading academic research centers, industrial partners, nonprofits, and government agencies to answer core questions about how the human immune system fights disease. As of this week, there are over 3.7 million cases worldwide of SARS-CoV-2, the virus that emerged late last year to cause the COVID-19 pandemic. This global emergency requires unprecedented efforts for prevention and control of the pandemic. As part of our response, the Human Vaccines Project, in partnership with FHI 360 and the Center for Infectious Disease Research and Policy at the University of Minnesota, has established the COVID Vaccine Initiative. Our initiative aims to ensure the development of safe, effective, accessible vaccines for the most vulnerable to severe disease from COVID-19. The CVI's focus will be on accelerating vaccine development and deployment for aging populations and those living in low and middle income countries. Excuse me. As part of the initiative, we recently launched the COVID Report, a weekly newsletter to provide a synthesis of key issues and data central to accelerating vaccine development. And today we are pleased to welcome you to the first global COVID lab meeting. The lab meeting will provide researchers with a neutral forum to share their findings and innovative ideas with scientists from around the world. By sharing and discussing the research frequently with a global community of experts, we're hoping to increase the likelihood and speed of identifying solutions toward ending the COVID-19 pandemic. The lab meeting will take place bi-weekly starting today, and the session will be recorded and available on our website and our social media channels. Our first speaker in this series is Dr. James Crowe, Jr., who is the director of the Vanderbilt Vaccine Center and the Anne Scott Carroll Professor of Pediatrics, Pathology, Microbiology, and Immunology. His laboratory studies the human immune response to infection for a variety of major human pathogens, including influenza and many emerging infections. He's authored over 350 scientific papers and is a recipient of numerous accolades and international awards. Today, Dr. Crowe will talk about his studies on the identification and development of human monoclonal antibodies for SARS-CoV-2, which ultimately may become a treatment that could provide immediate immunity for first responders. The Crowe Lab is one of the leading groups working on antibody identification, and his team is planning to start clinical studies early this summer for SARS-CoV-2 therapeutics. During the talk, you can send us questions using the Q&A function in Zoom, which I'll collect and ask Dr. Crowe after his presentation. We'll have about 25 minutes for questions and discussion, and we really hope that you'll take the opportunity to ask Dr. Crowe your questions about the data presented today. It's now my pleasure to introduce Dr. James Crowe.
Great. Well, thank you for having me, and glad to be here and share part of the morning with you. This has been an extraordinary time, obviously, for the infectious disease and vaccine and antibody community. It's been personally disruptive to me and my group, just like it has been for you. Many of us have been quarantined in one way or another, or working from home while trying to execute and respond to the pandemic. So it's been an amazing experience, and I just want to share with you some of what the team's been doing here at Vanderbilt, and also with a consortium of partners that we have trying to make a rapid response. And our focus is on antibodies, human B cells and human antibodies, and so that's what I'm going to focus on today. The foundation of this and our association with the Human Vaccines Project has to do with a broad investigation of the human repertoire. So we had termed this the Human Immunome Program, and we are trying to determine the sequences of all the B and T cell receptors in the human population, which was a really broad and ambitious goal. You'll remember the genetics of antibodies are quite complicated. There's 51 or so variable genes encoding heavy chain, and then there's about 27 diversity or D genes and 6 J genes, and then there's allotypes of these, polymorphisms, and these all recombine into VDJs that have junctions, an N1 and N2 junction. And so you get an enormous complexity. When we started, the theoretical complexity here could be something like 10 to the 18th heavy chains or antibodies in your repertoire, and you don't have more than about 10 to the 10 B cells in your body, or 10 to the 11th lymphocytes and so on. So you can't have 10 to the 18th at any one moment, but the theoretical possibility repertoire was enormous. And we launched in to figure out how many antibodies do people have and do they share them and so on. The genetics is important. If you look on the right to structure, this is a representative FAB, the antigen binding portion of the antibody, and there's heavy chain on the left and light chain on the right. And the principal determinants of interaction are the complementarity determining regions, CDR1, CDR2, CDR3 of the heavy chain, and then 1, 2, 3 of the light chain. And the CDR3 of the heavy chain tends to be longer. It's a lot more diverse in sequence because it's encoded by the V, D, the J, and the junctions. So really you get most of your specificity typically from the CDR3. But the whole sequence is interesting because you can get somatic mutations throughout. So in the human immunome program, we were leukapheresing individuals, collecting as many as 40 billion white blood cells, which come in bags. I mean, literally bags with weight to them. We separate cells and put out hundreds of vials with tens of millions of cells. And then we comprehensively sequence those, the heavy chain, light chain, and actually the TCR alpha and beta. We've been doing T-cell receptors as well. And you see an enormous complexity. We started getting views of what the human immunome actually looks like. So the preliminary views are that each individual in their blood at any one time has probably about 20 million clonotypes. So that's defined as a V and a J in the heavy chain with a particular CDR3. Now you can have somatic variants of those as well, but there's basically 20 million particular antibodies or lineages in your blood at one time. We've looked at people now year over year, 3 years in a row, and there's a substantial conservation of a person's repertoire from year to year. And then we look between people, what proportion of the immunome is shared between people. If you look at the heavy chain and B cells, about 3 in 1,000 or 0.3% are shared between individuals. And on the T cell side, we've looked at CD4s and CD8s. They're more conserved actually. So this is very exciting because that means you and I share a significant number of clonotypes, and therefore, if we're vaccinating, it'd be really interesting to know what is the diversity of clonotypes that are responding to any particular infection, whether it's flu, SARS-CoV-2, or whatever. And really for vaccine rational design or monitoring, you'd like to know what are the conserved ones, or what are the ones that we share, because you'd love for your vaccine to target the shared ones. Therefore, that vaccine might be more effective in more people or broader proportion of people. And then we've been studying the repertoires of at birth in cord bloods, healthy adults, and even supercentenarians, people over 100 years of age, to look at the change of the global repertoire over life. And what's interesting there is we've seen public clonotypes that are present at birth, they're in all adults, and they're in people that are 100 years of age. So there are some conserved and shared elements of the human repertoire that are there your whole life. And so I think this is sort of the fundamental basis on which we've been defining repertoires. So that's kind of a global basis. If you look at individual clonotypes on the x-axis, these are the top 100 clonotypes by size in 3 individuals, 1, 2, and 3. And on the y-axis, I'm showing you the number of somatic variants in thousands for that particular lineage. So each one of these would be an individual clonotype. And I just mentioned you have 20 million of these. Some of these clonotypes have 20 or 30,000 somatic variants. So when you make an immune response, you not only have tens of thousands, if not hundreds of thousands of clones capable of responding to that particular agent, but that each one of those can diversify into dozens, hundreds, or even tens of thousands of variants. So the complexity of the human immunome and its ability to respond to particular agents far exceeds the variability of an RNA-dependent polymerase type virus like SARS-CoV-2. So people wonder, are these viruses going to drift away beyond the limits of the human capacity to recognize them? And there's no way that's possible. The diversity that's possible in human immunomes is enormous and far exceeds the microbial space. So so we have used that type of technology and knowledge to start looking at individual infections, not just the whole immune system, but what about responding to one particular microbial threat or one pathogen. And we've, in particular, been focusing on emerging infections or pandemics because they're inevitable. We've already lived through, our laboratories lived through news cycles with H5, avian flu, H7, through chikungunya, Zika, Ebola, now Corona. It is just like clockwork. We can plan about every 18 months, we're going to be scrambling really fast to respond to something. So we've thought strategically how to address epidemics and pandemics in a more organized way. So we essentially have two ways of doing this. First is, can we go really fast? So something pops up, SARS-CoV-2, we've never seen that particular virus strain before, or even that, you can see a new species of virus arise. So you're not prepared for that, and so you need to move fast. And we've been developing fast platforms in what's called the Pandemic Prevention Platform program of DARPA, which is a defense program. And we got a large federal grant to develop the technologies to find antibodies within 60 days and deploy them. And I'll show you that we essentially can do that now. The other program that we have within our group, we've termed AHEAD 100, or Advanced Human Epidemic Antibody Defenses. And that's what if you thought, there's only a couple dozen virus families that cause epidemics. And if you look at individual viruses within those families, there's approximately 100 that you say, these clearly have the capability to cause an epidemic or a pandemic because they're present in animals, the vectors are around, like mosquito-borne viruses, arboviruses, are present in many places. There's an animal reservoir, and there are case reports or even outbreaks of these viruses crossing over to the human population. So people start making lists. Wow, these are all the viruses and other pathogens that we're worried about. So as an example, you could talk about flaviviruses. Flavy is yellow, so yellow fever virus-like viruses, flaviviruses. So we already know dengue is in 150 million, in 150 countries, 400 million infections a year. So we know flaviviruses can get around. Yellow fever is one of them, is currently causing outbreaks. Zika, big outbreak. Japanese encephalitis virus. In the US, we've had outbreaks of West Nile virus. And you could go on and on. There are other flaviviruses that are available to cause epidemics. So we've been making lists. The NIH makes lists, category ABC. CEPI in Europe, which is an epidemic preparedness program, they made a list of 8, and we conflated all those into a single list of 100. And we've been chipping away at these methodically, and I would say we have best-in-class lead antibodies for about 30 of those already. And to some extent, nobody cares because they're not currently causing an epidemic, but we're predicting they're going to care someday. So let me just give you some views of what the rapid response looks like. Last year, we did a demonstration. It was sort of a fake outbreak. We were trying to reduce our pipeline practices into something that could be done in less than 90 days. That was the original goal, and we called it a sprint. So we used Zika virus as a model outbreak. We'd already worked on Zika virus, so this was not happening during the Zika virus outbreak, but we simulated what if a flavivirus occurred and it was Zika virus. So we took immune cells, or PBMCs, from an immune donor on the first day, and we thawed them. We labeled them with antigen, which in this case we already had, which is not always going to be true, but we had antigen, and we sorted cells in containment. This is Pavlo Gilchuk, and the group was, against my own instincts. They were so confident that they could do this for the first time that they were tweeting this stuff out. So I'm just going to show you tweets from last year that were live fire tweets as they were doing what we call a sprint. So we're sprinting from blood to the cure. And so day 0, sorted cells on a cytometer, recovered individual cells or bulk cells, and then we make libraries for the gene sequencing, like I talked about in the Human Immunome project. We have all that data capability and pipelines. And so we set up libraries using liquid handlers, and an individual technician in this workflow can do enormous work with the liquid handlers. We got those sequences sequenced using next-gen technologies. We sent them to Twist, which is a company that does high throughput gene synthesis. They ship them back, and then in 96-well plates, we do transfections. So we do many expressions, and we can do at least 1,000, if not several thousands of antibodies at one time, just with 2 people. So this is Rachel Nargi and Pablo Gilchuk, who together expressed all of the approximately 800 antibodies we made at one time. And the idea is we either start with plasmablast or antigen-specific memory B cells. You put them into some kind of single cell encapsulation. I'm on auto advance for some reason, but I'll fight through that. And in this case, we were using 10x genomics technology, sequence, look at bioinformatics, synthesize, express, and then do some sort of in vivo delivery from protein from CHO cells or mRNA. We did both of those in parallel. Mike Diamond is our collaborator at WashU for this, and they did mouse protection studies on the fly of 20 lead antibodies, and they did a series of mouse studies because these antibodies were so good, we were getting complete protection in a lethal model of Zika all the way down to 0.25 milligrams per kilogram, so which was extraordinary. And we did this both as IgG delivery and as an mRNA delivery, in which we give the genes for the antibody and the mice make it. And then we progressed to non-human primate challenges in macaques. If we give the Zika antibody the day before challenge and then look at viremia, there's absolutely there's no virus detected, whereas the sham animals, they shed virus. This was done in Dan Baruch's lab at Beth Israel. So what we were able to do was to go from blood sample to complete protection of non-human primates in 78 days. So that was sort of our simulated outbreak. And this really involves a pipeline of technologies. It's not one single trick, but trying to do things that are more qualitative and higher throughput. So we knew that we could move fast, and lo and behold, we were getting ready to do another simulated sprint. We were going to take another organism and say, if an outbreak occurred, boom, here's day 0. And right while we were planning, a real one happened. And this is a New England Journal paper describing, January 19th, a man showed up with 4-day history, the first U.S. case that was known for a long time. Their cases now being identified in autopsy and so on. But this is in January, this case occurred. And on day 8 or 9, we had contacted the team in Seattle and worked with the investigators there, and they sent us a sample. And I think this was day 10 of infection of the first case in the United States. FedEx delivered to my home on Sunday morning a sample that we tracked from Seattle, and we took it to the lab and started. So we got into this, as early as one could possibly do. Fortunately, others in the field were doing very important work. Jason McClellan and others have provided structural information on the spike protein. We know the spike binds human ACE2 as a receptor with high affinity, and there's a fusion event with conformational changes. And we already knew that neutralizing, and in fact non-neutralizing antibodies, but particularly neutralizing anti-spike antibodies for other coronaviruses prevent from infection. So all that background information, we use that to hypothesize that human antibodies to SARS-CoV-2 spike would be neutralizing, especially if they block binding to human ACE2. And so again, these are structural information that's already in the PDB, and you may be aware that the spike's a trimer, but there's some dynamic features. So the spike can be closed, but you see in the middle there's a more open form. So the trimer is opening, and then the interaction of the receptor binding domain, or RBD, with human ACE2 has been defined in the residues, which we'll show in orange in this presentation, are the human ACE2 binding motif. So you would predict antibodies sitting up on the receptor binding domain in blue, and particularly contacting the orange region would block attachment. And in fact, that turns out to be true. And so in this case, we used antigen labeling and sorting, as I described before in the other sprint, and we made full-length S protein, really based on the work of others, in which the pre-fusion S protein was stabilized with some prolines. We knocked out the fusion peptide, put a trimerization domain, and so on, and made this in mammalian cells. We also made just the receptor binding domain itself. So if we label cells, sort them, and then activate them, and then we took them to a microfluidic device called a beacon, which is made by Berkeley Lights. And Berkeley Lights has been a great partner. They dropped an instrument on site and worked with us to do this work. And I'll show you how this instrument works. So the general scheme is fairly straightforward, but we had to build these tools on the fly while we were doing the work and validate them, not with pre-existing reagents, but with the patient samples we were getting, which was a little aggravating because you're developing the positive controls as you're isolating them. But basically we use a streptavidin bead and then biotinylated receptor binding domain or spike protein, and then we made recombinant human ACE2 as a reagent. And if you put streptavidin with biotinylated spike on, and then the human antibodies in blue that we're screening for bind to those beads, you can detect human IgG or human immunoglobulin of any type. We actually find different isotypes bound. And so in the single cell format, these are called pens. So there's a little slot. It's a very small volume, and one cell is put into each of these pens, and that single B cell secretes antibody that fills the pen and starts spilling out into this channel, and the channel is filled with these beads. And so you start detecting at the top of a column, it's like a smokestack or like a volcano. You can see antibody is labeling these beads, meaning the B cell at the bottom of that pen is making a spike full-length protein-specific antibody. And here, this is RBD, and the RBD beads are in green. So a single B cell making antibody labels the beads up at the top channel. So we were able at a single cell level to visualize binding to spike or RBD, and then you can actually recover those single cells off the instrument. And beyond binding, the team developed essentially what is a mimic of a neutralization assay. So if you take streptavidin and load it with RBD, receptor binding domain from spike protein, and then you add human ACE2 and an antibody to human ACE2, then you can see human ACE2 binding to RBD. And this purple antibody was able to bind to RBD, but did not block ACE2. But in some cases, this yellow antibody is binding directly on the contact site, and it blocks binding of human ACE2. So in a bead-based assay, we essentially have a receptor blocking assay, which is a surrogate neutralization. So essentially, we're doing neutralization on single B cells in a single cell device, and visualizing them as live cells, capturing them, and getting the genes out. And here's another view of that. You can put beads or B cells single into these pens, and this is an RBD binding assay. Again, the volcano from the bottom, antibodies coming out, and at the top you see binding to RBD. And then if you do the ACE2 blocking, in this case, the bead is fluorescing because RBD and ACE2 are binding, so there's ACE2 on that bead. But in this well, there's no fluorescence because the antibody from the single B cell is blocking the binding to the bead, so there's no human ACE2 RBD binding. So this is just an example of how they did the surrogate neutralization assay on single cells. To mitigate the risk, we were actually developing that assay and using the beacon instrument for the first time as we were doing the experiments. In parallel, we did the same workflow that I mentioned in the Zika sprint using a 10x genomics Chromium workflow, and they also were very helpful, dropped us in a new instrument because, if samples if the patients are viremia, that work has to be done in a BSL-3. So 10x Genomics helped us out by, we were already 10x Genomics users, but gave us another instrument to expedite this and containment as needed. So PBMCs, enriched, put in single cell, gene sequence on Illumina NovaSeq, get lots of sequences. We have a team who do bioinformatics on the fly. This is all automated workflows, and they curate it. And then we decided the antibody leads to take forward. We sent them to Twist, depositing directly onto their synthesizers in California electronically, and then on the cell phone say, go, we're ready to go, and they'd hit the button, make hundreds or thousands of antibodies. They sent them back, we expressed, did binding and neutralization. And I just, it's amazing the throughput. So at any one time, these are shade cultures of individual human monoclonals. And this is Rachel Sutton and Rachel Nargi in the cold room doing purification. So this is a protein maker instrument. And you'll see in this case, Rachel's actually purifying 24 human monoclonals of SARS-CoV at a time, and they can do this on this instrument milligram scale so that we can do mouse studies and structural studies on the fly as we're developing these things. So at every step of the way, stuff's super high throughput. In order to do neutralization at high throughput for a virus for which there had never been a neutralization assay, there weren't even reagents when we started. We used the same type of neutralization high throughput assay that we had developed in the Zika sprint. And this is using an instrument called an Intelligence, or made by ASEA, and it measures cell impedance. So on the x-axis, you have time, you have increasing signal for impedance. So as the cells are growing on the instrument, they make a nice monolayer, and you get more and more impedance. Then you add virus. And this is cool because you don't even have to have any reagents. You just have to have the virus. It could be unlabeled virus. You don't even have to know what the virus is. You can put a virus in, and then the cells continue for a period of time, a few hours, to increase their impedance. But if there is no inhibition of the virus damages the cells and the impedance goes down. However, if antibodies neutralize or inhibit the cytopathic effect of the virus, then the cell impedance tends to go up. So you can see the ones on the top are neutralizing, the ones on the bottom are not. This is just a qualitative screen. In most of the cases, we didn't even know the concentration of the antibody, but we were running hundreds of antibodies at a time, and this can be done in a 96 or 384 well format in live reading. So we transferred our device to WashU, to the BSL-3 or one of our devices. This is Brett Case, and Swati Srihari, among other people in the Diamond Lab. So we decided not to use VSV pseudotypes or pseudovirias to do screening because those do not give you accurate IC50 values for neutralization. We just use the live virus Wuhan strain, and these guys are spending long, long hours with BSL-3 for many reasons, but among them running that Intelligence neutralization assay. Once we identify hits, we go back and do larger scale monoclonal expression. We're doing, again, all this in CHO cells. And now we have purified antibodies of known concentration coming off of chromatography, as I showed you. And then we're doing either binding assays and neutralization assays that are quantitative. And this is just a shot that the team gave me in which we're doing 4 parallel ELISAs. The top left is full-length S. The top right is just RBD. The bottom left is SARS, or SARS-1, sometimes people call it. So we're looking at cross-reactivity. And the bottom right is the N-terminal domain of SARS-CoV-2. And you can see we get a lot of hits. Some of them are RBD, some are NTD, some are neither, meaning they bind somewhere on full-length S that's not RBD or NTD, or at least not represented in those reagents. Could be the stem, could be quaternary. And then a subset of them are cross-recognizing SARS-1. And interesting, so the take-home is anything that binds SARS-1 is probably not neutralizing. So you've seen a lot in the literature and in the news about old SARS-1 antibodies that cross-react, and there's crystal structures, and a lot of people talking about, but the ones we've seen don't neutralize the the older ones that pre-existed, and in fact, the ones we're getting that are neutralizing for the cross-reactivity is not a good marker for neutralization. In fact, it's probably the other way around. And then Diamond Lab is doing quantitative neutralization with purified antibodies, and you see, I'm just showing you two of approximately 1,900 antibodies that we've looked at, and we get potent antibodies. So the next steps really are animal studies, and we have animal studies ongoing or have been completed in 2 or 3 models, and ultimately they'll be in 4 or 5 different models that people are developing. We're doing EM and crystallography within the lab. We already have structures of some of these, so we know where they bind, and their binding epitopes are consistent with their activity. For instance, the potent neutralizer ones are often directly onto the receptor binding domain as expected. We spent a lot of time trying to get these out to other people, so we've done options and licensures to manufacturing partners, and I think 3 of these are already announced. There'll ultimately be 4 or 5, 6 companies developing some of our antibodies because we made so many of them, and we have so many potent neutralizing antibodies. There's a lot to choose from, and there are different patterns and phenotypes. And many of these partners are using different strategies. So some are using Chinese hamster ovary cells, which would be standard in the industry, but this takes time. One group is using plant cells to express the antibody, which is actually quite fast, but ultimately harder to scale for world supply. One of the groups using mRNA, in which we did a first-in-man study with a company, one on an mRNA company last year, in which we made a chikungunya antibody, converted that to a sequence and gave them the sequence, they made the mRNA, and this company was Moderna. They put the mRNA for a chikungunya antibody into people by IV infusion, and they express the antibody in their body. So you don't need a bioreactor. Your body is a bioreactor for mRNA. There's other people have been using DNA, like Inovio does DNA for this purpose. So there's different delivery methods, and we're providing to various people who have different ways to do this. We've already sent out a large number of antibodies to manufacturing partners to make diagnostic kits, and we have pairs of antibodies that are very sensitive for detection of protein or virus, and these are being incorporated in rapid antigen tests that hopefully will be rolled out in the next few weeks. Also the antibodies are important for vaccine development because we know that many of them recognize conformational epitopes, not linear peptides, and therefore vaccine companies are interested in acquiring these antibodies for use so that they can characterize their vaccines, or their vaccines recapitulating the conformational dependent protective neutralizing determinants. And once they get that working, not only can they validate their general vaccine construct is good, but then they use them in a lot-to-lot comparison, and they're using it as a release reagent so that they can say this lot is as good as the last one or is as stable and so on. So these antibodies are already being used for diverse purposes, and wow, so many people were involved in this. This is a photograph of my group at Vanderbilt. Not everyone, just like your labs, not everyone's even allowed on campus. On the left are the people that have been touching the SARS-CoV-2 project, and there are more than this, but Rob Carnahan's running the group as he did the Zika sprint, and Pavlo and Seth are really the core individuals who are designing and executing most of these really difficult assays. Naveen's an immunologist in the group who's been doing a lot of the work. Andrew and Joe are running a lot of the genetics, sequencing, synthesis. Rachel, I showed you their video. They're spending a lot of time in the cold room making proteins. Elad, EM expert. Lauren Williamson is doing competition binding assays. Nurgen Kosei, in parallel to everything I told you, has made hybridoma-derived antibodies, and those are in process. Jinwei Dong's doing crystallography. Ryan Irving's running the lab as a manager. These folks are bioinformatics. as well as our human subjects and regulatory team. We work with Berkeley Lights. I mentioned their collaboration and also their instrument, WashU. We've also done with work I didn't show you today with Barrick Lab at UNC using alternate neutralization assays, and they're doing some of the animal stuff. Beth Israel, Dan Baruch's lab, I showed you primate studies. Eun-Young Lee at Emory also has participated in the work she has discovered media that sustains single B cells and plasmablasts in culture. And she's been a good collaborator. And University of Washington, Helen Chu has been a clinical collaborator with samples. Mario Ostrowski in Toronto also. I didn't mention all of the subjects that we've studied were infected in China and traveled to North America. So they've been good collaborators to identify those subjects and obtain samples. And then the funding has come from DARPA P3, the NIH. Dolly Parton made a major gift to Vanderbilt to support this emergency work. And we received a Future Insight Prize last year from Merck to do pandemic response. And then I mentioned the instruments. And also, all of the human immunome work I mentioned has been done under the Human Vaccines Project. Wayne Koff, Ted, Sabrina, who's running our webinar today. And these are all the folks, including companies who help support that effort. And our friends in La Jolla helped us with that. So I hope that was helpful, and I'm happy to take questions.
Jim, that was great. Thank you so much. We're getting a lot of questions. I'd like to start, we're getting a couple of questions about the clonotypes. So you mentioned before there are 10 to 20 million clonotypes. Are these just derived from blood, or does this include naive cells and cells in tissues? And there was a question about the conserved clonotypes, if they have something to do with innate immunity rather than acquired immunity.
Yeah, those are good questions. I think the work that we've done at scale, doing billions of sequences, was all done on leukopaks from blood or large volume blood draws, like 300 ml blood draws in the subsequent years. We have looked at tissues, not at scale. And when you do look at body tissues, I think we've looked at 12 to 15 body tissues. The tissues are different from the blood in that there are a lot of naive cells in blood, and there's very few naive B cells in tissue. If you just look at the sequences, they tend to be mutated, they're class switched, and so on. So the tissues are definitely different from the blood, and that's a whole nother frontier I think we need to take on, is doing deeper sequencing of tissues. That's difficult. Just technically, it's more difficult to obtain billions of tissue resident lymphocytes and do those. And the other thing about the field is it's really moving to heavy chain, light chain linked, or TCR alpha and beta linked, which is done with a single cell RNA-seq technique like 10x genomics or some kind of water and oil emulsion technique. A lot of that's evolving. And people ask us, well, why don't you do them linked in the billions? And currently you cannot afford, or you cannot technically do the linked immunomes in the billions. A 10x chip will give you, 10 to 80,000 shots on goal if you're lucky. That does not get you to tens of millions. So there's not a great way to do linked repertoire. So I think right now, before we do too much large-scale immunome, we're trying to harden off the tools a little bit better. The immunome we've done is mostly out of blood. And then what was the second question, Sabrina?
Sure. The second question was about conserved clonotypes and whether that has to do with innate immunity versus acquired immunity.
Yeah, so we're very interested to know the antibody or the receptors that are there in everyone. In fact, pre-exposure, they're there in cord blood and so on. They must be doing something. They must be there for a reason, because they're overrepresented to a degree that you would not expect. They could be appropriate to bind to many antigens of a certain feature, structural features or charge. We don't know that. So what we need to do is synthesize and express thousands of these and then pair that with an expression library. And there are people in the field who have made every human gene an expression library, all microbial genes, and we need to probe our libraries against the microbial and human structural genomics expressed libraries to figure out what they're binding, if anything. Another possibility that we have thought about is that actually they're not involved in defense per se. So not even innate immunity, but they might be involved in physiologic homeostasis. So let's say you have a cell in your body and a B cell comes along and binds to a receptor and gives a little bit of a signaling, a constitutive level signaling. That could be an autoreactive or autoimmune interaction that's actually beneficial to that cell, because now it's still humming with a little constitutive signaling to keep metabolism going. And therefore, it may be that T and B cells are walking around touching all your cells just to keep everything going. And that would be essential that everybody have those. And I'm just waving my hands. I'm just bringing up that they could be selected for conserved threats. They could be for biochemical features like charge, or they may have some other non-immune role. But we just don't know. So those are great questions and future directions.
Yeah, so speaking of future directions, it's a good segue. We're getting a lot of questions about the next steps for the antibodies. How do you envision the antibodies being used therapeutically? Would an IV infusion be expected to work in a lung environment, and what mechanism? And if you could just talk a little bit about the timing of upcoming clinical trials and studies, that would be great.
Okay, well, there's three basic ways that you could use antibodies. One is prophylaxis before you're exposed. So we have people working in the ER, in a hospital, or first responders. Those sort of people, we could give antibody. If we put a 90-day or longer half-life extension FC mutation in there, we might be able to give coverage for 3 to 6 months for prophylaxis, which essentially is what we want to do with the vaccine, is protect people for a period of months or years. So probably we can provide immunity to people who have high risk exposures as prophylaxis, and bridge to a time in 1, 2, or more years where we have a vaccine. The second is called post-exposure prophylaxis. So you go to an event, it turns out somebody there was infected and exposed you. You may or may not be infected. You don't have symptoms, but given the transmission rate, you have a high risk of already being infected, and we give antibody there. That also likely would work very well because the virus titer is probably very low. You're not having symptoms, and so the antibody could prevent disease. And if we can just keep the peak titer low in that setting, the disease corresponds to a large degree to the peak titer. So if you post-exposure prophylax people early during their infection, interrupt the curve and lower the peak, you'll have less symptoms, and that should work. The third is therapy, and therapy is going to be very complicated. Most antivirals for acute infections don't work because by the time people show up, they're already over the peak of their shedding, and the shedding is going down, and T cells are kicking in, and bad stuff's happening with cytokines. So it may be in a setting where an antiviral small molecule doesn't work, an antibody may also not work because an antiviral effect is not sufficient. So you might envision an immunomodulatory where you have an anti-cytokine and an antiviral antibody. That might be the key to therapy. So those are the three uses. And then in terms of distribution, prophylaxis is almost certainly going to be sub-Q or IM. It's not going to be IV. And we can give up to about 30 milligrams of protein to a typical adult by sub-Q. There's even ways to give 100 ml sub-Q with enzyme additions. So prophylaxis, most likely going to be sub-Q or IM. If we're doing treatment or somebody who's exposed and it's high risk, IV would be fine. And you can give a lot of antibody IV. In flu programs, people have given 3.5 or 4 grams of antibody pretty commonly. So you give high dose IV. And then the question about biodistribution from serum into the lung, it's already known, at least in animal models and in human BAL studies, that you can achieve something like 1 to 100 or 1 to 300 of the serum or body fluid titer of antibodies in the lung space. So it's not a 1-for-1. The titer you get in the serum after passive administration is not the same as in your alveolar space, but 1% of a very high titer is probably going to be protective. And for other viruses, this works fine. You can give antibodies to flu and completely protect or clear the lung. We already have licensed antibody for RSV. So the principle should work. It's just there's a gradient from body fluid to lung. And the gradient is fairly steep. It's about 0.3 to 1% of the levels in your body fluid are in the lung.
Okay, great. I'm seeing a lot of questions now about workflow and preparation for other labs who may want to take this work on. Everyone is saying your workflow and operation is very impressive, obviously, you know, very a great setup over at Vanderbilt. What are the hurdles for continuing to decrease the response time for developing neutralizing antibodies for an unknown future outbreak? And for this outbreak, how would other labs scale up and get ready? What reagents are needed? What are the preparations that other facilities should seek out?
Well, first of all, the biggest bottleneck right now is DNA synthesis. So all the antibodies I showed you, I think, or most of them came from a sample we got March 17th or something like that, 14, 15, 16, 17. So 3 weeks later, we already done thousands of antibodies, neutralization, down selected, competition. We can do the whole thing in several weeks. But once we get the genes and send them to Twist has been a great partner. I mean, they really are integrated into our workflow, and they've done a lot of innovation around DNA synthesis with us and for us. But still, you're looking at a 9-10 day window there. So of our month, 9 or 10 days is just the DNA synthesis, which is aggravating. Wow. So I think figuring out how to go directly from a single cell to expression is, I think, a desire that we have. In terms of other labs doing this, we're trying to miniaturize, and we're also trying to teach people how we do this. We have a paper that hopefully will be out shortly about the Zika sprint that shows in great detail exactly how we did this. On the other hand, this is very, very challenging. I make it look like it's easy. We just bring in a blood sample and rip out 2,000 neutralizing antibodies. It's not that simple. I think the complexity of just the compulsion of doing everything properly, quickly, cold, the organization, and we didn't get here in one day either. I think I've been working in this field for 25 years, and over the last, I'd say 3 to 5 years, we've built a LIMS system, a laboratory information management system. We built databases. We have our own supercomputing cluster. We have our lab is set up in terms of cores. We have genomics core, we have protein expression core, and so on. And furthermore, the instrumentation alone, I mean, we probably have $10 million of instruments here with liquid handlers and all that. So, I'd like to say that we are gonna teach people how to miniaturize and expedite this stuff, but at the scale we're working, there are not many groups that are gonna be able to compress the time frame, because it's not just working quickly. You got to work at scale, because the antibodies are stochastic. And I didn't really mention, but we expressed something like 2,000 antibodies over the last month, and we ended up with more than we need for multiple companies. But really, you're talking about 30 highly potent antibodies out of 2,000 antibodies. So are you prepared? And I don't think that's a fault of ours. That's the biology of the virus. Many of the antibodies, even to close conformationally correct spike, are not neutralizing. So are you prepared to work at that scale? And so, if you made 100, you're going to get 1. And is that good for you or not? So I'm not certain I could recommend these workflows for most labs, but anyway, we'll be teaching people how we do this stuff, and again, shortly publishing a paper that's very clear in terms of all the methodologies.
Great. We're getting a lot of questions about the ADE response, so antibody-dependent enhancement. Can you talk a little bit about the risk for ADE and how you rule that out, and just your thoughts related to COVID-19?
Yeah, this is a complex area, which could be an entire seminar, so I won't be able to do it justice today.
Sure.
So antibody-dependent enhancement has been developed as a field, mostly in flavivirus, dengue. So antibodies to the dengue fusion loop for one serotype from a primary infection may, the hypothesis is, because they cross-recognize dengue viruses of another serotype, they may tag that virus, not neutralize it, leave it as a replicating virus, and then the FC binds to an FC receptor on a cell, which is permissive for infection. So like a Trojan horse, you're taking an infectious virus into something like a macrophage where more virus comes out, and more virus is a bad outcome. And it's known that rarely, something, like 1 in 1,000 of people having a second dengue infection will have severe disease, which is considered enhanced disease. So in vitro, you can show more replication in a macrophage line or something like that. And in vivo, you see enhanced disease. And the question is, are they connected? And people are trying to work that out still in dengue. And again, that's an entire field, whether T cells also participate in that, and what types of antibodies do or don't do that. And will every antibody do ADE in vitro? The answer is yes. Typically for dengue, if it's at a subneutralizing concentration in particular assays, they'll do ADE. But does that actually happen in the body in a polyclonal response? So ADE is very complicated in terms of enhancing FCR. There's another aspect of ADE that's been talked about in flu stem antibodies, in which it may be that some antibodies trigger a conformational change, which normally would be neutralizing, but in the right kind of timing, you could hypothesize that you could enhance fusion and make viruses fuse better or faster or at a time when you wouldn't expect them, and get more flu disease. And I think that's very controversial in the flu field. It's only been seen in certain animal models and not in others, and not always reproducible. So that's being worked out in the flu field. Does ADE occur? We also did Ebola work where we're certain in small animal models we can recapitulate ADE happening sometimes for Ebola. So, but does that happen in humans? Hard to say. So now we're to SARS-CoV-2. There are some animal papers with other coronaviruses, prior work suggesting that prior infection with a non-protective response will cause enhanced disease with a second infection. And this has been attributed possibly to antibodies. So this is going to have to be worked out very carefully. I don't think you can just say, I have an antibody, I have an assay with a macrophage-like line, and I put them together, and I got more RT-PCR signal. Oh, that's bad. We're going to kill people. I don't think that's responsible to interpret those assays, but that's the type of thing that needs to be initiated. Also, maybe with primary cells, and also epidemiologically, we need to understand, does this even happen in nature? And then finally, what would you do if you thought it was a concern? So they're not our group, but there are other groups working on antibodies in the field. One group's turning up the FC activity because they think it's important to function. And there is evidence in SARS-1 and MERS that NTD, non-neutralizing antibodies, protect. And if they protect, they're probably doing it through FCR-mediated mechanisms, and you probably want to tune it up. So some people are turning it up. Some people are just leaving it wild type. Some people are knocking out the FC interaction with FCR in their therapeutic candidates. And there are mutations in FC that you can introduce to abrogate interaction. So there's a spectrum of opinion and activity going on with ADE. And I don't know how it's all going to play out, but that's how you can deal with it, is either turn it up, leave it alone, or knock it out.
Okay, interesting. Okay, so I'm going to take one more question and then we'll wrap up. Given the zoonotic transmission of coronaviruses, we were just talking about the different animal reservoirs, the bat to pangolin to human, is it feasible to predict with artificial intelligence and develop pan-coronavirus cross-reactive neutralizing antibodies for pandemic preparedness? Is that something that would be a feasible approach?
Yeah, I think if you go back to our ahead 100 idea, sure, you can make 100, but what if there's 101 and it turns out to be another corona, another flavy, another phyllo? So within each project, like we're doing SARS-CoV-2, we're also currently looking at SARS, MERS, HKU1, 229E, OC43. We're looking at cross-reactivity. The problem so far is any antibodies that we've seen that have been cross-reactive are not neutralizing or very weakly neutralizing. So this suggests that there's some structural differences or kinetic differences in exposure of SARS-1 and SARS-2 and the other viruses. So it may or may not be possible. I think we're going to press on because it may be the receptor binding domain is not the only place where you can get a cross-reactive response, and maybe neutralization is not the only protective response. So what if you had somewhere else on the stem that was conserved across all these viruses, but was also protective? And so we're pursuing that idea. And I think it is desirable, because if you currently had an antibody that inhibited and protected against all known coronaviruses that we had, and I just gave you a list, the likelihood of a new one falling under that umbrella is high. So I think it's a great idea to look for cross-reactivity, but it needs to be coupled with protection. So that's the way we're thinking about it.
Great, thank you. Well, I'd like to take this opportunity to thank Dr. Crowe for sharing his insight and knowledge on COVID-19 with us today. And thanks also to the attendees for your participation in this discussion. We got a lot of questions. I'll have to see, this is our first webinar, so I'll have to see after the fact. I think we get a transcript of all these questions. I'll try and relay back and forth, and we'll try to respond to you after the chat. I'd also like to invite you to our next session. It's 2 weeks from today in the Global COVID Lab meeting, which is scheduled for 10 a.m. on May 21st. Our next speaker is Dr. Bet Korber from the Los Alamos National Laboratory, and she's going to be speaking about her paper that has recently been making the news on recently identified mutations in the spike protein of SARS-CoV-2 and the impact on vaccine development. If you have any questions, please feel free to write to us. My email is up there on the screen. And if you have any suggestions for future speakers, please also email us, and you can follow the conversation on social media using the hashtag #HumanVaccinesProject. Thank you for attending, and we look forward to seeing you at the next lab meeting. Dr. Crowe, thank you so much.
Yeah, thanks for coming, everyone.